our speaker, Dr. Jennifer Braswell. A few words about her. She is a senior advisor at the ARM Foundation for Cell and Gene Medicine, having held senior leadership roles in regenerative medicine for the past 15 years. Her career has spanned academic and clinical research, development, training, and nonprofit management at the Sanford Consortium, the Sanford Stem Cell Clinical Center at UC San Diego, and Army BioFab USA. Dr. Braswell is a catalyst to successful collaboration. She sees relationship building as the key to success for public, private, governmental, and nonprofit partnerships to advance cell and gene medicine. So I'll welcome Jennifer. We're so thrilled to have you, and I will let you go ahead and share your screen. And while Jennifer is doing that, let me just say, the science here is complex. It's confusing and can feel overwhelming. And I'm 5 years into working in this field. And as a non-scientist, I still don't fully comprehend a lot of it. So, research studies show that one of the biggest concerns people have expressed about gene therapy is not having appropriate information or being able to make an informed decision. So we know that you are getting questions about cell and gene medicine from your communities, and we want to equip you with some answers. So today, Jennifer is going to give us a primer on cell and gene medicine, and we hope you'll join us again for part 2 of this series at our Rare Leader Meetup tomorrow to learn how to now, once you are armed with this information, to carefully and sensitively transfer it to your communities, and we'll give you some tools to help you do that. So, how you doing, Jennifer? Are you set up?
I think I'm set up. I'm about to turn my video on, and you should be seeing now my first slide.
Okay, it looks good.
Okay, good. Thanks very much, everybody. I'm really excited to present this because my effort throughout my career has been as a liaison between the hardcore scientists and other people who are trying to know what they know. Learning at the right level, I often say we go from what we do know to what we don't know. So the intention of this presentation is to give the basics on cell and gene medicine so that you can start integrating this understanding into what you really do know. A little awkward with the technology here, but I want to tell you that I'm the senior advisor at the ARM Foundation for Cell and Gene Medicine, and Healing Genes is our education program, the flagship education program of the ARM Foundation. I'm really grateful to Global Genes for giving me this good forum to present information. And one of the things I'm particularly interested in is getting feedback about the parts you think I effectively communicate. So I'd encourage you to work with Global Genes to express your feelings on that. What I'm going to talk about today is understanding genes at the very basic level and understanding cell and gene medicine as its own field, talking about why it's different than drug development and implementation of traditional medicine in the US and in the developed world. I'm going to talk about how cell and gene medicines work. And at the end of this show, there's a list of approved cell and gene medicine and some future tips for you. This goes over a little bit about the ARM Foundation for Cell and Gene Medicine. And I want to point out that the way this slideshow is designed, in the future, it will be made available for you to customize for your own use. And I'm hoping it presents in a way that you can internalize it and then use it yourself. What I like to talk about when I talk about cell and gene medicine is to get early on the terminology and the location of all the parts of the gene, cell, tissue, and the body really well fundamentally comprehended, because it will help you understand how gene therapy in particular and how cell and gene medicine is supposed to work. So the illustration is just to get all the pieces in the right place. Your genes are made up of DNA. Those are the molecules that are making up your genes. those genes are located on the chromosomes, and the chromosomes are tightly bundled inside the cell nucleus. It's the DNA in the genes that contain the instructions for our cells to build proteins. And this instruction guide to building proteins is the way the genes tell our bodies how to work. So genes influence everything about the way bodies work. The other things you need to know about them is they come in pairs, and we typically get two versions of each gene, one from each of our parents. But versions of the genes in general in the human population are widely various. So genes have a number of variants from just a few to quite a lot. Some genetic variation, that is, some of the varieties that a particular gene come in, don't make any functional difference. And my best example is eye color and hair color. A variation of the gene for the genes for eye color and hair color can send the message to make the eye color of the iris different color, or the hair have different texture and color. But that genetic variance makes no difference in the functioning of the iris or of the hair. But sometimes the genetic variation means a gene is not built correctly. And these small adjustments to the DNA sequence in a gene, the sequence of these building blocks, is called a mutation. And the terminology I'd like to share here is this idea of the genetic code. You hear it a lot in the news and you hear it a lot everywhere, but it's sometimes hard to understand what the code is referring to. The DNA is made up of only 4 chemical building blocks, and they're known by the letters A, C, T, and G. And these letters, or the building blocks themselves known as by the letters, are arranged in long strings on our genes. And it's the string of letters that scientists and doctors and people familiar with the field refer to as the genetic code. And the mutations are small adjustments in those codes. Sometimes they can be larger adjustments, but it's the pattern of those letters that are in the DNA that are then wound up into the gene, wound up into the chromosome, wound up into the nucleus. And if the genetic code is defective or missing or a piece is in the wrong place, that's when the proteins are made incorrectly. So now, from the story of all the genes, I'll start to talk about specific genes and when specific genes can be defective. And when they're defective, they don't make the protein correctly. And this can happen if the environment affects a baby's gene before it's born, and the environment is then causing a mutation. A baby can inherit one defective version of a dominant gene from one parent. A baby can inherit 2 defective versions of a recessive gene from both parents, or there are changes to the entire chromosome. And since chromosomes carry a lot of genes at once, this affects many genes and the makeup of many proteins. So when we try to understand how disease is caused, many rare diseases are caused by a malfunctioning gene or genes. And some of the most serious incurable diseases are caused by genetic defect. And that's because as the body grows and the cells divide and differentiate, the erroneous gene keeps being copied over into further and further cells. And this slide is one that you've probably seen pictures, but this is that are similar to this. These are pictures of how the genetic code has been modified. So on the left side of the screen, for example, you see an example. The first is of a point mutation. And you see there that string of letters that represents the way DNA is encoding the instructions for the creation of the proteins. So there's a few examples given where those DNA building blocks are inserted or deleted. Then further errors can be caused when a gene is duplicated or inverted. And then when the whole chromosome is involved, you can start to understand that a lot of genes are being altered at the same time. So at the present time, it's single gene disorders that effective forays from research science has been making great inroads. So this is not a comprehensive list by any means, but the single gene disorders are the ones that have been currently most effectively pursued, cure has been most effectively pursued. And this raises the point about how disease is usually treated versus what cell and gene medicine hopes to do. Prescription pharmaceuticals are typically used to manage the disease, to mitigate the symptoms, relieve pain. But typical pharmacological courses of medicine can be disrupted by a lot of things. Treatments can start and stop because of other things that are happening in the life of the patient, other illness, but also just lifestyle issues. Furthermore, of the 7,000 rare diseases that exist, the vast majority have no current treatment. Diagnosing, although it's getting much better with the advent of a number of both scientific and political actions, diagnosing is getting better, but it's still difficult. And the traditional course of treatment usually involves a very, very big team, as you well know: physicians, pharmacists, teachers, household assistants, and so forth. Cell and gene medicines, in contrast, look to target the cause of the disease. So if treatments are effective genetically, the patient should no longer have his or her disease. The hope is that the treatment would be a one-time effect, and the subject diseases are those rare inherited disorders that are of significance to a huge part of the population. Another point about cell and gene medicine, however, is that gene therapy is targeting somatic cells. That's all the cells of the body, excluding our reproductive cells. So if a treatment works for the patient, it is not expected to be inherited and in the next generation. So as I've said, there are two main challenges with the way disease has been treated traditionally. One, there's very few treatments for genetic mutation diseases, and conventional treatments only target the symptoms. So what I've done is I've outlined the areas of cell and gene medicine I'm going to talk about and describe here. You'll notice that there's 6 things on the left side, and there's only 4 things in the orange circles. That's because this field is very new. There's a lot of terminological overlap, and words are sometimes grouped together in different ways by different explainers. But I'm going to explain gene therapy, gene editing, tissue engineering, and I'm going to start with cell therapy. I got my slide out of order, but many gene-based diseases might be approved by cell and gene medicine. And I'm putting up this list because I want to point out where I know about a lot of active research. It's, again, not an exclusive list. There are researchers who work on areas where their heart and scientific interest has led them. And I just want to mention also that there is cell and gene medicine being pursued for non-genetic disease, certain cancers and osteoarthritis, which is a plague to many people. So cell therapy involves transfer of cells to the patient who is suffering. And the idea is that the cells are not working right or absent, and the cells that are put in will grow, replace, repair the other cells. It's easy to understand cell therapy if you think of the most common kind, which is blood transfusion. In blood transfusion, the cells of whole blood from one person, red blood cells, white blood cells, lymphocytes, other things, are moved from one patient to the next. Blood transfusion, thank God, in my life is a commonly used safe therapy. It is actually just before my life began that it was even thought to be possible at all. This is an idea that gives me a lot of hope, since blood transfusion is now quite common. Bone marrow transplant is another cell therapy. Cells are that contribute to the formation of the blood are moved from one person to another. So we usually talk in cell medicine, but with two particular words that I'll try not to use too often, but you'll hear them. One is allogeneic. That's what I have described, something like blood transfusion. You're receiving cells from someone else. Autologous, on the other hand, means that you are receiving your own cells back. These words are so commonly used that it's important for you to know them, but the concept is going to be the same no matter how it's referred to. Because genes reside in cells, both cell and gene medicine are using cells. So again, we've got these building blocks we've built up from DNA to the gene to the cell. And stem cells, which you've heard of, have two characteristics that are important. They can divide and make more copies of themselves, and they can divide and become different types of cells. And I'm going to go quickly through ones that you've likely heard of in the news and just tell you a little bit about what you need to know here. Pluripotent stem cells are the cells that can become almost all the cells in the body. And usually we talk about embryonic stem cells that are from leftover embryos that result from in vitro fertilization, creation of embryos, and induced pluripotent cells that are adult cells, some somatic cells, sometimes from skin or blood, that are genetically reprogrammed to be the same sort of cells as embryonic stem cells are. Bone marrow cells and blood cells contain hematopoietic or blood-forming stem cells. This is a kind of stem cell that can form all the cells in blood. Mesenchymal stromal cells are much in the news of late. They're isolated from bone marrow, adipose or fat tissue, and other tissues. They have the ability to reduce inflammation and perform other positive things. And you will be hearing about them probably as you hear news about suppressing lung inflammation in COVID-19. Cord blood stem cells are another kind of blood-forming stem cells, and there's wonderful work in that area. There are also cells used in anti-cancer immunotherapy. I'm going to just show them on the slide here. And then there are further cells that are used in other stem cell treatments, such as retinal pigment epithelial cells that replace and repair stem cells in the retina, neural progenitor cells that give rise to neurons and are being tested for diseases and trauma of the spinal cord, and pancreatic islet cells, which work well in treatment of diabetes type 1, but are in short supply. So there are researchers working on replacement pancreatic islet cells. Gene medicine and gene therapy is a subject of great interest, I know. This is where the healthy genetic code that I've referred to, literally the string of those letters on the building blocks, are put into the patient's body with the idea of preventing disease expression, reducing a disease's severity, reducing further damage caused by the genetic disease, and they have the potential to cure the patient of the genetic disorder from which they are affected. And this is an important slide. Don't be alarmed by the illustration on the left. On the right, I'm going to walk you through it. Gene therapy works by replacing a mutated gene with a healthy gene. But the important thing is, we've got to get the healthy gene in across the barrier of the nucleus into the nucleus of the cell, so that the cell can create the appropriate protein. And my point here is that as you've heard all the confusing and elaborate science of gene therapy, the point is that researchers are choosing the right approach based on the cause, the genetic cause of the disease. So they're trying to understand at a deep level the functioning of those genes so that they can create the right therapeutic. How am I doing on time? Okay, good, thank you. So there are two ways to create a treatment that are going to correct the malfunctioning gene. One is in vivo. That's going to be a direct delivery of the corrected gene to the patient's body's cells. We call that in vivo because it's going into the living body of the person. Then we have ex vivo treatments. That's when someone's cells are going to be taken out and a newer corrected gene is delivered to the cells outside the body, and then those cells are reintroduced. So you can see in each case, I want to be talking about how the DNA of the genetic information is getting into the cell so the cell can produce the right protein. The thing to understand is that the doctors need a way to get this gene with the corrected information into the cell where it's going to be accepted and incorporated somehow into the chromosomes that are so tightly wound up in the cell nucleus. The mechanism they use is a vector, and the vector is the delivery vehicle. Very often, the vector is created from a virus. So you'll hear it called a viral vector. You'll hear a lot of talk about the virus. The vectors, the delivery vehicles, are made from parts of virus. Virus works in the natural world by having a very efficient mechanism of giving its genetic information to cells of its host. So that ability of viruses is used to now carry the new desired genetic code across the cell barrier and into the nucleus of the cell. So different types of viruses can be used as vectors. And this is, as I said, the situation where the research and medical teams are choosing the appropriate one to use based on how the genetic disease operates, both at the cellular level, but also in the level of the whole person. So adeno-associated virus, you'll hear a lot about. It can insert its genetic material into quite a number of cells. And the words maintains a high level of gene expression over time. That means that the genetic information can make the cells make the proteins correctly for a long time. Lentivirus is another kind of virus that's it's well understood and it's used sometimes. It integrates permanently into the nucleus of the cells. So sometimes that is a desired effect, sometimes that's not desired based on how the disease functions in the person. Adenovirus, which is similar to adeno-associated virus, but it's a different type of virus, can hold large segments of DNA. So when that's wanted, that's the kind of virus used. And retrovirus operate in a very specific way, transferring their genetic information with RNAs. The point is that there are a number of kinds of virus. That is not the complete list. And that's what you will hear a lot of detailed information about from the science teams. Gene-modified cell therapies are those therapies that I talked about I mentioned a second ago, where cells are taken from the body, modified ex vivo, and put back into the body. CAR T-cell therapies against cancer are the best known of these gene-modified cell therapies. And there's a little bit of terminological difference between who uses gene-modified cell therapies and who calls this cell therapy and who calls it gene therapy. But if you've got the building blocks of the science well understood, it shouldn't be confusing. Gene editing aims to correct that genetic code at a specific location in the DNA. Gene editing is a great advance of just the last few years. And this slide is meant to, the picture is showing you a picture of a socket wrench over DNA. The idea is only to communicate that these techniques like CRISPR-Cas9, TALENs, those are tools to cut the DNA in a specific way. So again, an illustration you might see about how this works shows those building blocks. And hopefully by now we understand that those are the letters that represent the genetic code. And the illustration is supposed to be showing that these nucleases, these specific chemicals, can be used to cut the DNA of the gene in a very specific way. And that's why we call it gene editing. It's as if there was a piece of paper with those letters written and the nucleases cut the part out, and then a natural process repairs it. And it's the repair that changes the genetic information. I'm going to talk one slide about RNA medicines, only because it's a very important area of early research. And if I know the scientists and doctors, they will be harnessing these techniques. There has been one RNA therapeutic approved so far. Tissue engineering now brings a whole lot of these techniques to the foreground and uses genes, cells, tissues, which are a variety of cells that work together, and sometimes whole organs, manufacturing them or cobbling them together so that damaged tissue can be replaced. Sometimes you hear the word biomaterials, which usually refer to non-human materials that are used to replace tissue. Regenerative medicine is another term commonly used. That's now become an encompassing term that takes under consideration all the different methods that I've talked about for regenerating cells, tissues, and organs. This is the list of cell and gene medicines approved in the United States by the United States Food and Drug Administration. You can see the list there over at the left-hand side. I hope that in the next 5 years, we will have a much greater list. And I think that's likely because there are gene and cell medicines approved in Europe that are being, approval from the FDA is being requested now. cord blood products are also listed there. Cord blood and the storage of cord blood is also regulated in the United States by the FDA, and that's an important thing to understand. I'm gonna suggest that you start to visit healinggenes.org, which is our website, for trusted, vetted information that it is my overwhelming goal to make understandable. The thing that I want to achieve is to have concepts well understood by you and terms comfortable for you to use so that you can engage with your doctor and with other people, and have a good understanding of the information you need to ask and a good understanding of the answers you're receiving back.
Jennifer, could you go back to that slide just really quick?
Sure.
And we can just explain to people that first bullet point has in brackets, insert presenter's website or
Yeah.
resources from the presenter. So that's an example of what we'll be providing to our Foundation Alliance members, right? So that they can include their own information for their disease community.
Exactly. I hope that the slides that I've presented can be used easily by Foundation Alliance members. And the slideshow is designed so that you can take slides out, and also so that you can put your own name and your own information into the slide presentation for the benefit of the groups that you care most about.
Right, and that's why it's so important that you give us your feedback on this, friends, because, you know, we want you to find this helpful, and we want you we want to equip you to help educate your communities. And so based upon what you saw today, this slide deck, this presentation, you know, let us know what you think. If you felt like you learned something, if it was helpful, if it was consumable, we want to hear, you know, all of that feedback, so we can help the ARM Foundation continue to refine this. And we're going to talk more about, again, tomorrow, you know, equipping you to teach your communities tomorrow during that Rare Leader Meetup.
Absolutely. Thanks, Suzanne. This is a list of additional organizations from which you can learn more about cell and gene medicine for rare disease. A number of them are highly, highly technical. Others are easier to understand. What I'm also trying to do is present information in a variety of ways so that your way of learning is available to you. So if you like to watch videos, you can learn from videos. If you like to read things and absorb, if illustrations make an impact on you, all of these are presented. And then I need to thank everybody who not only helped me make this presentation, but throughout my career, it has been patients and their advocates who have most formed both my motivation and my desire to understand the science.
Let's switch gears for a second, Jennifer, and try to address some of those commonly asked questions and even some questions that have come through the registration page from this event. So one thing we get asked a lot, and this was actually the topic of one of the summit sessions that was recorded yesterday: how do I know if cell or gene therapy is an option for our community? Can you talk to that?
Yeah, so it's a complex question to answer, because the answer, it depends, is always so unsatisfying, but I'll try to give some broad parameters. The application of cell and gene therapy, especially gene therapy, is most suitable when first of all, the disease or the ailment, the series of ailments are well diagnosed. So that's the most important thing, that something has to have been diagnosed as a genetic disease. And next would be as I said in the show, the single gene disorders are at the present time the most amenable to gene therapy. Um, and uh, the the next thing is that a treatment, either an experimental treatment in a clinical trial or an approved treatment, has to exist. And the reason I make this point is that there's a lot of science news about discoveries for rare and genetic diseases. And sometimes it's the news is presented at a very early stage in research. So gene therapy is right for you when the treatment is actually available. And your doctor is the place you'll find out about that.
Right, right. Okay. All right. Many people ask, how can I get a genetic test?
And that's not a good one for me to answer because my knowledge set is in basic science and in genetic testing is a little bit outside my domain, but I know that it's through your care provider that this would be available to you.
Right, right. I would say ask your provider. There's definitely different types of genetic tests, and Global Genes has a toolkit that describes some of the most common. So you could go to globalgenes.org and search our toolkits under the search term genetics, and you'll find that toolkit. And you can also find I do know you can find a genetic counselor at nsgc.org.
Oh, good.
nsgc.org. So that would probably be helpful for folks. Okay, so this question I hear all the time. People hear the word virus and viral vector, and they are concerned that the virus that's used in their body might infect them. So, so can you help people understand, will the viral vector infect them? And another kind of follow-up question that we get asked is, what if I have antibodies to the viral vector? What happens then?
Yeah, good point. Viruses have been around a long time before we knew to be scared of them. But we also know that major non-genetic diseases are caused by virus, like influenza. And so it's natural to get sort of triggered by the words. But one of the things that I'm hoping to do is empower people to understand that the same thing that makes, and I'm threatening to drop into the technical word, but pathogenic viruses, meaning bad viruses, it's important to understand that scientific study of how bad viruses infect us is exactly what is making the ability to create tools from virus, because they have the ability to carry this genetic information and be the vector. So the virus used in gene therapy is extremely unlikely to infect you because it's not it's a disabled virus. It doesn't.
Yeah, it's dismantled. Yeah,
it's dismantled. It's a better word. It's taken all apart, so that it is not a virus that is carrying the genetic information that makes you sick. And then you were asking about whether you have antibodies already for the virus.
Right, what happens then?
Yeah, so what happens then, and that's part of the "it depends" answer. I was mentioning before that researchers and doctors are engineering their therapy specifically for the disease they're trying to cure. So there's a lot of work so that the common viruses that you would have built up antibodies to from being a person in the world are evaded. It is possible that in the course of your natural life, you've developed an antibody that will attack the vector. But frankly, this was much more of a concern about 5 years ago when the treatments were less developed. Now the ability to evade that effect is has been perfected by people who are getting their treatments approved by the FDA. That's exactly the sort of thing that the FDA needs to see evidence of. They need to see evidence that the treatment will be effective in the people who have the disease.
Got it. Okay, thank you. And questions we get often are around how long the therapy will last, how long the effect of the therapy will last. Is the cure?
Yeah.
And I know that we use the term durability, and in your presentation you talked about gene expression over a long period of time. So can you expand on that durability concept?
The doctors talk about a durable effect to just mean it lasts a long time. It's durable. So it is the goal of gene therapy to affect a change in the body that lasts for the lifetime of the person. The truth is that we don't know enough because we haven't treated enough people who have lifetimes for us to compare. So the design of the therapeutics are made to either be durable or to be of shorter duration. And again, that's how the gene therapies are being designed and use the different kind of dismantled viruses as the vector to treat the disease appropriately. So for some diseases, you do want to suppress the expression of a protein and then see what happens when it comes back. So when someone is exploring and learning about a therapy for their own disease, that issue of whether it's supposed to last for a lifetime or not is quite clearly stated. That's exactly the sort of issue one should ask one's doctor if they're considering an approved therapy or participation in a clinical trial.
Okay, that's really helpful. So can you talk just for a minute, we got about another minute or two left, about, what can you tell people about life after gene therapy? And is a whole lot of follow-up required? Are you monitored for a long time to come by your healthcare team?
This is a huge topic. It really is. And the answer to the second part is yes, you should be expected to be monitored by your care team for the rest of your life. Absolutely, yes. And my heroes who are participants in clinical trials, part of their heroism is their perseverance and their dedication to having their experience benefit other people. There is no better definition for me of a hero, because it's not what you risk when you are ready to risk it. It's what you do the rest of your lifetime to help everybody else. So yes, that's an aspect of life after treatment. But there are a number of others, and you and I have talked socially about this a little bit. We're aiming for medical treatment and amelioration of disease, not miracles. So what you should expect is the changes that have been explained by the doctor and research team, but not the kind of thing that you see sort of a cartoon character rising from a wheelchair, that kind of thing. There are things that improve muscle function, and rising better than you've risen before might be one of the expected outcomes. But the changes to your life and your lifestyle are dramatic we hope are dramatic in the good ways. For example, there are a number of gene therapies that are aimed at suppression of proteins that the current course of treatment is constant administration of oral medications, including like throughout the night, that you have to awaken and take more medication, and then awaken a few hours later and take more medication. So one of the changes that might really be a great benefit is the ability to just rest comfortably during the night for the whole night and wake up and start your day. But yeah, there will be a lot of changes to your life.
Yeah, that could be an endpoint. It's a quality of life measure versus necessarily that your disease is fixed.
Mm-hmm.
And, you know, I think we all acknowledge that this regenerative medicine, cell and gene medicine, you know, these transformational medicines have so much promise and so much hope, but we have to manage people's expectations too. I mean, you know, to understand, you know, a lot of questions that are still being answered about this, and that this is not going to be a therapeutic approach that is available for everybody.
Yes.
Much is being studied and learned about it right now.
Mm-hmm. Mm-hmm. Yeah, and all the questions that are arising in our discussion are absolutely the things that should be asked of the healthcare team and the research team. But furthermore, the feedback from patients, caregivers, families, and everybody, teachers, household assistants, everybody involved, has really informed the scientific community about what amelioration of daily life should be an aim. And that's been a giant contribution. When people talk about what would make their life improved, sometimes, for example, improved continence, improved muscle tone, easier breathing, which is could be a step towards a cure rather than going for the moonshot. This has been a huge contribution of patients and their families.
Right. Well, that's all the time we have for questions, but we encourage you to you know, email us at advocacy@globalgenes.org, and we can do our best to get those answered. Thank you so much, Jennifer.